So we're beginning even more new things at this point, and we're talking about the cell. So to give you a brief introduction, in 1665, Hooke examined a piece of cork with a crude microscope, a real old one. I'll show you my 1930s microscope, and it's pretty crude. But he identified these little rooms as cells. Leeuwenhoek, Hooke, who was working about the same time, used a really refined lens to describe living cells from blood, sperm, and pond water. And since the days of Hooke and Leeuwenhoek, they've improved microscopes and have vastly expanded our view of the cell. So we get to see some of these incredible images. Isn't that amazing? I'm impressed. Okay, so in this lecture, I'll tell you this. We will definitely do this one. Oops, let me draw a little bit better. And then we're going to have some options for how to do this one and this one. Okay, but these, at least three kind of big ideas are what we're going to cover by the end of our little time here. So introduction to the cell is we're looking at what structures. Let's see if I can spell this properly. What structures are in the cell? And what are their functions? So common theme in biology and most of the stuff we do is structure and function. So this one's going to be a huge one about structure and function. And I will tell you, you have to focus on the organelles and their functions, because the STAR test loves that one. Oh, they love it. Love it. Okay, so that's just a helpful bit for you guys. All right, so a variety of microscopes have been developed for a clearer view of the cellular structure. So the first microscopes were the light microscopes. And I'll show you the one that I have, which is amazing. Because it still works. Not well, but it still works. It's from the 1930s. But visible light passes through a specimen, through a glass lens, and is finally projected onto the viewer's eyes. That's the one that we're going to be working with as well, but ours are a little bit more updated. Specimens can be magnified up to 1,000 times, and some of the ones that I've used at the college can do up to about 4,000 times. So we'll take a look at it. Magnification, so these are some good terms, is an increase in an object's size compared to its actual size. Okay, so it's making it appear bigger. Resolution is the measure of clarity of an image. So in other words, it's the ability of an instrument to show two nearby objects as separate. So magnification, it's like when you're touching the screen of your phone and you're trying to like push it out so you can see it closer up. That's magnification. Resolution is like when you're moving your phone around and you're trying to wait to get a clear image and you're waiting for your phone to focus for a second. That's the resolution. Phones, good example here. I'll put it here for you. Phones. All right, so microscopes do have their limitations though. The human eye and the microscopes have limits of resolution as to how easily you can tell two things apart. So therefore, a light microscope can't provide details of the small cell structure. It can give us some important things, and we'll actually practice this. Microscopes are a really, really important thing to note, so we will make sure that we have time. I'm trying to work it into 2 separate days, so we have plenty of time to work with the microscopes and get comfortable with them, because that's a skill that we need as biologists. Okay, amateur or whatever, we need the microscope skills. So using light microscopes, scientists studied microorganisms, animal and plant cells, and stem structures within cells. I used a high-powered microscope back in the day, so I don't know if you know what an aphid is, but it's about about that size, about those little 3 dots together. And I had to use a microscope and a very, very tiny set of like tweezers, which are called forceps, and take the guts out of the aphid. Yeah. Okay, you think this step's boring. You think about getting your eyes all crossed because you're looking at a microscope trying to take the guts out of a tiny insect all day. You think I have sympathy for you? I do not. Just kidding. Anyway, so in the 1800s, these studies led us to this cell theory. All right, it states that all living things are composed of cells and all cells come from other cells. There's sometimes a third component. Why is it not in my brain? I think it's just that basically the cell's the smallest unit. So if you, oh my god, my pen. Smallest. I'm trying to dig that out of my brain, but sometimes the whole point of this is sometimes there's kind of a third bullet to the cell theory, and that's fine. These are kind of the big two that we can deal with, but there is kind of the third part, and you guys could Google it and check it out, and if you're like, miss, you're way off, that's cool, just tell me. We'll get it clarified in class. But the cell theory is one of those big ones. Just composed of cells, cells come from cells come from other cells. They don't come out of nowhere. Here's a beautiful image of this looks like to be paramecium to me. Oh, this is a better one. This is a scanning electron micrograph of paramecium, so you can see all of its little cool hairs. He's one of our protists, which we'll talk about more when we do classification next time. So he's a cool dude. This is what's called a transmission electron micrograph. So they're a little bit different. This one's a composite, so it's both transmission and scanning. So an electron microscope, though, because I thought I was going to talk about those. Oh, I am. Excuse me. We'll go through some of these a little bit later on, but just to see what you can see with your eyes is you can see humans. You can actually see a paramecium in a human egg. It gets harder after that. You need a light microscope to see about really from the paramecium down to about the mitochondria level. Beyond that, you're going to need the electron microscope, which is what started in the 1950s. So scientists started using a very powerful microscope called the electron microscope, and it views these ultrastructures of cells. So instead of light, it actually uses a beam of electrons and just like pummels this thing with tiny electrons. So it can resolve biological structures as small as 2 nanometers. I'm pretty sure if we do the math on, you guys have had chemistry more recently, but I'm pretty sure that's 2 times 10 to the negative 9. That's tiny. It can magnify up to 100,000 times. And for the bug lover like me, this is such a cool thing. So scanning electron microscopes study a detailed architecture of their surfaces, whereas transmission will actually go into internal structures. I have a friend of mine in Florida who is now a professor, and she's doing research on all kinds of cool things, but she got the contacts for the USDA people right nearby who have a scanning electron microscope. I told her this is a nerdy moment in me. I was like, wow, I'm so jealous. I would so love to play with that machine, because they're not like the little microscopes we're going to look at. You can't pick them up. You can't carry them. I don't remember the number, but there's a low number of them in the US. So they're pretty rare. And so it was very exciting. And so it was like, give me contacts, let me play with it, but not today. Differential inference light microscopes can amplify the differences in intensity, so we get some different structures within them and get that 3D. So that's what we saw before. We're at about 9 minutes, so even though we're doing these things in class together, I always encourage you guys to pause, take a break, look over your notes, stretch a little bit. If you got to stand up, I won't look at you like you're a weirdo, okay? If you really must, go to the bathroom. If you got a snack, discreetly eat it, whatever, and then come back into it. All right, so cell size must be large enough to house the DNA. We're going to get all into that. The proteins, all those structures that we need to survive and make more cells. But they have to remain small enough to allow a surface to volume ratio that will allow adequate exchange with the environment. We're going to do a demo with this one a little bit. I won't have you kind of do it because now we're a little shorter on time. But there are some cool aspects of it. So like if we take kind of the same block, but if we do it in different units, if you do the actual volume of this block, it comes out to 27 units cubed. The surface area of it will be about 54 units squared. So this kind of surface to volume ratio comes out to 2. Whereas if we take these same little guys, the same block, and break it up into these chunks of 1 unit, we'll end up with a surface area of 162 units squared, which gives us a 6 to 1 ratio of surface area to volume, which means it's a lot easier to get things into the center. What did I do? There we go. It's easier to get things into the center than it is from here. We're going to look at that and demo it so you can kind of see what I'm talking about. I'm going to keep my pen working. Okay. As we go along, there are some structures that we got to make sure that we know, and this is going to be one of the first ones. So the plasma membrane, or you'll hear it also the cell membrane or cellular membrane, things like that, actually forms a flexible boundary between the living cell and its surrounding. It's going to contain phospholipids. Those are those things that we talked about in Chapter 3, and they form a 2-layer sheet called the phospholipid bilayer. So there's hydrophilic heads, and they face outward, which is exposed to water, and hydrophobic tails. So the heads kind of look like this in a row, and we're going to do a bilayer real quick. And then they've got these hydrophobic tails, and they're kind of these little chains, and they look like this. And the thing is about these little phospholipids, they always look like little jellyfish waving at each other. Look at them, they're waving at each other. Hey, buddy, that's nice to meet you. Alright, so these are hydrophobic tails, and these two here are the hydrophobic heads. So you can see kind of here, water would be here, and water would be here. Hopefully you catch my drawing on that one. If not, we'll go over it more. We're going to have a whole chapter dedicated to what it does. So membrane proteins are embedded in the lipid bilayer. Some proteins form channels that shield ions and other hydrophilic molecules as they pass through the center. We'll have some other proteins that serve as pumps, using energy to actively transport things in and out of the cells. So we will have membranes that are within that protein. We will have proteins within the membrane. Cross that up a little bit. Okay, so a really good demonstration. Inside and outside of the cell are both going to be water-based. Okay, so in and out of the cell are water-based. We do have these channel proteins, and for some reason your particular book just loves to make all proteins purple. So that's a thing. If you notice that they're kind of purple things there, those are definitely proteins embedded within that. The hydrophilic heads, as I showed you before, and the hydrophobic tails that don't get along with water are waving at each other and forming that protective boundary. Okay. It also goes to show there's some interesting parts to these proteins is that they're going to have regions that are hydrophilic, which it's trying to tell you is in here this particular, or hydrophobic. These are those regions that are hydrophobic, and then within that same thing there'll be regions where they're hydrophilic and get along with water. So a lot of proteins have different parts of them that are different structures to make them fit in there well. So something to think about. All right, this is another big concept. We gotta make sure that we know this one. Have to know it. We've got two different types of cells. So the first one that we have are prokaryotic cells. My goodness, did not draw that line like that. The other form of life are eukaryotic cells. So you could actually look this up. But the two are distinguished by a membrane-enclosed nucleus for eukaryotic cells, and then membrane-enclosed organelles. So I learned this in one of these lovely videos that I can show you guys, and you can see it a little bit later. But pro means before. Okay, and kary actually means kernel or nucleus. Okay. So nucleus or kernel, think like a corn kernel. So it's like before the kernel. And then U as actually means true. So kary again means kernel or nucleus, so they have a true nucleus. So the big thing there is eukaryotic. Eukaryotic, yes, they have a nucleus. And they also have membrane-bound organelles. Prokaryotic cells are going to be simpler and smaller too. So this is another good bit to know. Okay. Prokaryotic and eukaryotic cells have some common themes. So these are things that they have in common. I have to excuse me for writing large today. I don't know what. I need to switch out my styluses, I think. But anyways, so they have some things in common. So the plasma membrane, they're filled with a jelly-like fluid, cytosol. It's also called cytoplasm. So if you hear cytoplasm, that's cool too. Oh my goodness, I can't write. Look like a child writing here. Okay, cytoplasm is the same as cytosol for our intents and purposes. We're going to have some chromosomes or some kind of DNA, some genetic material in there, and then ribosomes. So these are the only things they have in common. 1, 2, 3, 4 things they have in common. Membrane, cytoplasm, chromosomes or DNA, and ribosomes. That's it. Okay? All right, so inside both types of cells is cytoplasm. So there we go. I'm getting a little bit more technical. So, however, eukaryotic cells, the term refers only to the region between the nucleus and the plasma membrane. We can go technical on those, but at the same time, if you use them interchangeably, I'm not going to get that bad about it. I usually just use cytoplasm. In a prokaryotic cell, here's some differences. So before the kernel, the DNA is coiled in a region called the nucleoid region. Just means like nucleus-like, nucleoid, nucleus kind of. But it has no membrane to surround that DNA. Outside the plasma membrane of most prokaryotes, there's a fairly rigid, complex cell wall. So in some of them, we're going to find this cell wall. That helps kind of protect shape and maintain it, but there's some distinctions on that. We'll get to those a little bit later when we deal with more kind of bacterial stuff. But know that there's going to be a cell wall there. Some prokaryotes have surface projections. So like short projections, like they project outward, they come out, help prokaryotes to help attach prokaryotes to each other and their substrate. You ever read a sentence and you can't just get it out? There's one for me. There we go. Longer projections are flagella, and they'll propel the prokaryote through a liquid environment. Okay. So the short ones are going to be cilia, and the longer ones flagella. We'll talk about those coming up a little bit later. These guys in particular will have some short hairs that are called fimbriae, but the big things here that we need to know is that there's ribosomes, there's a nucleoid space, there's a membrane, we'll have a cell wall there, okay? There's our bacterial chromosome, and that's about what we're looking for. We can talk about capsules and fimbriae, and that's all good, but these are the basic things that we want to get and be able to identify. And identifying their location is important. So like IDing where they are in the cell is good. It's actually really important. All right, eukaryotic or true kernel or true nucleus cells contain membrane-enclosed nucleus, boom, and various organelles, which literally means little organs, which perform specific functions in the cell. They come in kind of 4 basic functions, so we're given 4 general categories. 1, we have the nucleus and the ribosomes that are involved in genetic control of the cell. So we've got kind of the control centers with nucleus and ribosomes. We'll talk about the endoplasmic reticulum, the Golgi, lysosomes, vacuoles, peroxisomes, all involved in manufacture, distribution, breakdown of molecules, those kinds of things. They distribute, they break it down. Then we've got mitochondria and chloroplasts. They're going to be dealing with energy processing. And finally, we'll have structural support, movement, communication, all that stuff, which will be cytoskeleton, plasma membrane, and cell wall, and some. Okay. The internal membranes of a eukaryotic cells partition into compartments. So there are some compartmentalization within those cells. Cellular metabolism is just all the chemical activities of the cells, and it occurs within those organelles. Okay. Almost all of the organelles and other structures of animal cells are present in plant cells. There are a few exceptions, and so this is good to note plant and animal cell differences. especially we'll get to it more when we look at structures, but it might be nice here. So lysosomes and centrosomes that contain centrioles are not found in plant cells. I will say the lysosomes things might be a little bit debatable, but typically for our intents and purposes not found in plant cells. The only sperm cells of plants, a few plant species have flagella. I'm going to get that out. Mainly those are going to be kind of movers through water, some things like that. Okay, so a couple of things. Some other things about plant and animal cells. Another big thing to note here. Plants have a cell wall. So we're talking about plants here. They've got a cell wall with cellulose, that's sugar. They have things called plasmodesmata. They're just channels through the cell wall. Because if an animal doesn't have a cell wall, it's not going to have the channels through the cell wall. Okay. They also plants have chloroplasts, and they have a thing called the central vacuole. So a compartment that stores water and a variety of chemicals. All right. So some of the structures we'll end up looking at, and we're going to go through kind of each of these individually, kind of. Okay. And then this one's the plant one. So this is plant, and we'll look at some of those differences. And we'll define what those things are, what they look like, and where they are, and what they do. Here's the thing. I'm going to kind of give you guys the option moving forward here. So I'm going to continue to kind of talk about the nucleus and the ribosomes, but if you are tired of listening to me, which is fine, does not hurt my feelings, what I want you to do is to at least go find the note part of the live binder, and I'll either you get it. If I haven't given it to you, say, hey, I've gotten to the part of the lecture and you didn't give me the table yet. Go get the table. You can write your notes down however you'd like to. If you want to write your notes down before and then move them onto the table, that's cool. If you want to take them directly on the table, that's cool. But what I want you to do is to complete a table that really talks about each of these individual organelles and their function and where they're located. So that's what I need you guys to do for me. Okay, but how you choose to go about that is up to you, because that's the main purpose of these sections. You may choose, if you're going to stop listening at this point, which is cool too, I would read through the notes, because I honestly can't remember right this second if there's any other really, really key points that jump out. You may even choose to look somewhere else and just play this in the background, so if I say something that jumps out at you, that's cool too. Any way that you feel like doing this, do something, because our basic goal here is to get the organelle, what the structure looks like, and what the function is. And we'll review those kind of together in the next class period, but I want to make sure that you find them. That's it. Like, you can do it whatever way you want. If you want to do it on your own and not listen to me, that's cool. If you want to go along with me, that's cool too. But this is kind of the point, as to, like, you decide how you want to do this. You want to listen to me? Well, let's move on. You don't? Just stop it and go on. I promise I'm not going to put an Easter egg after this. I'm not going to mess with you if you're choosing to do this a different way. That would be unfair of me, so I'm not going to do that to you. There will be no Easter egg after this point if you're done with listening to me. Okay? All right, with that, we're going to keep moving forward if you're still with me. All right? Or if you wanted it a reference later, I don't care. Whatever. I'm just going to keep doing this thing because I'm sitting here. All right, so the nucleus contains genetic information. Big thing here. Here's our first one, the nucleus. It's got the cell's DNA, and the whole thing is it's going to be directing protein synthesis. Again, synthesis just means to make, so it's going to make proteins. It gives the controls, it gives the directions. It's really the direction sheet or the blueprint for making proteins. Now that word did not come out. I'm just going to cross the whole thing out. It gives directions. Okay, so that's a big thing. It gives the instructions, I suppose, is a better word. Okay, so and we'll go into that in one of those units, I think unit 4. So DNA is associated with many proteins organized into structures called chromosomes. We'll talk about this when the cell's not dividing and has some proteins with it. It's called chromatin because it looks like it says a diffuse mass within the nucleus. It's trying to tell you it looks like spaghetti noodles. Spaghetti noodles. Chromatin is going to be sort of equivalent to spaghetti noodles when we start getting there. So if you like that, get used to that. Okay. The next thing we'll have is a double membrane nuclear envelope. It has pores that regulate entry and exit of large molecules, and it connects the cell's networks of membranes called the endoplasmic reticulum. So it's going to connect with our ER that we'll talk about in a minute. But this is going to be the kind of the protector of the nucleus. It's kind of the fortress around the nucleus, keeping it safe. We do have a nucleolus or a nucleolus. I've heard it. Either one works. It's a prominent structure in the nucleus. So if it's here's the cell, here's the nucleus, there's a thing within it. I'll try to make it kind of dark here. But there's going to be kind of a dark portion, a part of it, and that would be the nucleolus. Okay, its whole point is to make ribosomal RNA, so we can shorten that to it makes ribosomes. I told you I'm having to write big today because my pen doesn't. My stylus is angry, and that's okay. We can work with that. Okay, so here's a better view of what that would look like. So you can see the nuclear envelope around the outside. You can see the nucleolus right here in the middle. You can see chromatin. See, it looks like spaghetti noodles. It's like a big bowl of spaghetti. Okay, and so that's our the whole bit here is the nucleus. And we're going to get into the endoplasmic reticulum, which I colored over there, but you can see them actually expanding out to becoming the endoplasmic reticulum. Okay, so ribosomes are the next thing that we'll talk about, and they're involved in cells' protein synthesis. So it just makes proteins. Ribosomes are the cellular components that are used to the instructions of nucleus, written in mRNA to build proteins. Cells have to make a lot of proteins, need a large number of ribosomes. So shorthand here, makes proteins. Boom. Can you understand that scribble? Boy, I hope so. Ribosomes are super important, and we're going to talk about them a little bit later. We do have some ribosomes that are free ribosomes that just hang out in the cytoplasm and the juicy stuff in the cell, and others are bound. So free ribosomes are suspended in the cytoplasm. They kind of look like those beads that are in some of the hand sanitizers. They're just hanging out in the juicy stuff. And then we have bound ribosomes that are attached to the outside of the endoplasmic reticulum. And some of the nuclear envelope, but basically the endoplasmic reticulum, or the ER for short. Okay, and so you can sort of see those here. We've got our endoplasmic reticulum, kind of the blue thing there, and it's these things that dot it. Okay, there's free ribosomes, and there's a bound ribosome. Okay, and basically we'll talk this whole process through. This will make sense later, but the big thing is they make proteins. Okay. All right, let's talk about the endomembrane system. Endo kind of means within, so we're talking about a system within the membranes, then kind of the endoplasmic reticulum and the cellular membrane. Okay, so many of the membranes within the eukaryotic cell are part of the endomembrane system. So these membranes are physically connected, and others are linked by tiny vesicles. They're just little sacs made of membrane, and they'll transfer membrane segments between them. So it's just the little moving pieces between them. Little vesicles act like little vehicles. Many of these organelles interact for synthesis, for making stuff, distribution, moving it around, for storage, and then for exporting or getting out molecules. So we're going to talk through the nuclear envelope, which we already have, the endoplasmic reticulum in a little bit more detail, Golgi apparatus, lysosomes, vacuole, and plasma membrane. So those are the ones that we'll cover. Okay, the largest component of the endomembrane system is the endoplasmic reticulum, or ER for short, and it's an extensive network of flattened sacs and tubules. Okay, so we'll have two kinds of ER. So there's the smooth ER that actually lacks the attached ribosomes. We have a rough ER that has bound ribosomes that stud the outer surface of the membrane. So the one we saw earlier, kind of like this, and it's got all the dots on it. If it makes it sort of look like it's itchy, it's a rough ER. It'll not have those little dots on it that make it look itchy if it's a smooth one. Kind of like that. Sort of. Okay, so we can sort of look at it here. This isn't necessarily the best one, but if you look over here, these guys are bound with ribosomes, the little brown dots. Makes it sort of look itchy. So we say that's the rough endoplasmic reticulum. The smooth endoplasmic reticulum is missing the little dots. So part of it, you can see sort of the vesicle here. They're both kind of involved in distributing some of the nutrients, so in the vesicles, and help distribute. Let's see, did I miss this part? Okay, I didn't. All right, so the smooth endoplasmic reticulum, its basic purpose is for production of lipids, oils, phospholipids, and steroids. So I'm going to say that's the biggest function of the smooth endoplasmic reticulum. It can also be involved in the production of enzymes that help, process drugs, alcohol, and other potentially harmful substances. It also can store calcium ions. It can do that, but the big thing is that it produces lipids, oils, phospholipids, and steroids. It's the maker of those things. The rough endoplasmic reticulum makes additional membrane for itself and secretory proteins, so proteins that will be secreted out of the cell. But the big thing here is if it's bound with ribosomes, it will make proteins. So the smooth one is making lipids, the rough one is making proteins. That's a very simple way to look at them. The Golgi is one of my favorite ones. It serves as a molecular warehouse and a processing station for the products that are manufactured by the endoplasmic reticulum. So products travel via vesicles to the endoplasmic reticulum and then to the Golgi apparatus. On the side of the Golgi stack serves as a receiving dock and transport vesicles produced by the ER. The big thing, you see, it's a warehouse, it processes, it helps kind of things travel. The big thing here, there we go, we'll add on to it, is that it'll help modify and process through its stack. It'll also do shipping. So the big thing with Golgi is think about it as a function. It's a lot like UPS, because it's going to take things in, it'll modify them or change maybe an address or a label or whatever, and then it ships it out. It says this is where you need to go off to other sites. So it's essentially a UPS. It's the mail. All right, so you can sort of see it looks like that. They always seem to look like it. There is a receiving side where it'll take things in, it'll modify it a little bit, and then it'll transport and ship out of the other side. Your little UPS station, the Golgi. All right, another good one is the lysosome, and it's a membrane-enclosed sac of digestive enzymes. So if it has digestive enzymes, okay, think about digesting things that we don't want within the cell. That's going to be basic job of cleaning. Okay. So think about the word lyso. Doesn't it sound like a different cleaner like that name? Maybe Lysol? Lysosomes are like Lysol. They're for cleaning. Now, it's made by the rough ER and processed in the Golgi, but whatever. So they fuse with food vacuoles and digest food, destroy bacteria engulfed by white blood cells, and fuse with other vesicles that are damaged to organelles and other materials to be recycled. It's Lysol. It cleans. You got to clean the whole thing up. Oh, we had a little thing that was about its formation. Maybe later. But here's the thing, here's like a lysosome in action. We have a little food particle. It comes in, it fuses, it digests it and breaks it down. And then here we have like a damaged mitochondria, and it says, oh, we don't need all that. And it'll break it down into little parts so we can use those parts over again. So it just helps clean up and recycle things. Okay? We're almost there. Vacuoles are kind of these large vesicles that have a variety of function. Some protists have contractile vacuoles that actually help them eliminate water. Plants have vacuoles for some digestive functions and pigments, and they have some poisons in them. That's pretty cool. If you can't move, you got to come up with all kinds of things to do. We'll try to look at this contractile vacuole and paramecium a little bit later. It's pretty cool to watch them just like squeeze in as they try to remove water. Pretty gnarly. Okay, you can see this one. You can definitely tell this is a plant cell because this is one of those things that they have that an animal doesn't, is a large central vacuole. Okay, so a review of the structures involved in manufacturing and breakdown. So we'll look at the figure. Okay, and so we should be able to kind of trace things out. But we start at the nucleus, which has our nuclear envelope and our chromatin, our spaghetti noodles. If we have something there, it will come out here with the bound ribosomes on the smooth ER. We can make some proteins. I think I said that wrong. On the rough endoplasmic reticulum with the dots, we can make proteins on those ribosomes. Smooth ER is going to make lipids. Ta-da! Okay, so then we've got proteins that are made in our rough ER and our ribosomes. Those little vesicles will go to the Golgi, who will end up breaking them, not breaking them down, but relabeling them to be shipped on the way out. A little UPS station. Our little outside part is our plasma membrane, which can take some things out. If some things are damaged or don't quite look right, we need to recycle them or clean them up. We can have a lysosome fuse with that vesicle and break it down and digest it, so we can keep moving forward. Okay, I think I got that right. Something doesn't sound right. You just send me an email and let's discuss it. All right, one last thing here is a peroxisome, and it's a lot like what you think it is. Think like hydrogen peroxide. Hydrogen peroxide is actually a naturally occurring chemical. Oh, that's my dog whining. Come here. And Charlie Horse here, that's my dog, has decided to join us for this lecture. The end of the very tail end. Okay, what do you know about peroxisome? Nothing yet. All right. Okay, well, let's get there. So they're a metabolic compartment that do not originate from the endomembrane system. We don't actually know how they're related to other organelles. But peroxisomes, so hydrogen peroxide anyway is kind of a naturally occurring thing that can help break things down. So we have some other peroxides that naturally occur. Again, they can break down some fatty acids and they can use as cellular fuel. So it's another sort of cleaner within the cell. Think about, you use hydrogen peroxide to clean wounds. That's kind of what these guys do, break stuff down, those fatty acids, so we have them as a kind of cleaner component there. Okay, so regardless of whether you're with me or not, this is where we're going to stop. This will be our next section, is to do energy converting organelles. So, you know, everybody hopefully will stop at this point in time. Okay, so we'll keep going with that a little bit later.